Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes, we have been discussing different next generation sequencing technologies such as 454 sequencing and Illumina. So, today we will be discussing one more technology which is called the single molecule real time sequencing, or in short SMRT sequencing, and we will see how this method is different compared to the last two that we have discussed so far. So, let us begin. So, this is the topic for today's class. We will discuss the single molecule real time sequencing method developed by Pacific Biosciences. These are the keywords that will come across. So, we will have 0 mode waveguides or ZMWs, we will see real time sequencing term quite often, and finally, we will also see something called hi-fi reads in the presentation. So, just to briefly recap what we have discussed so far, we have talked about 454 sequencing, and 454 depends on pyro sequencing, right. So, if you remember the principle of sequencing, we have this DNA polymerase, and when a base is added to the growing strand, there would be release of pyrophosphate. Now, this pyrophosphate then is finally converted to ATP, which in presence of the luciferase enzyme and luciferin will give rise to a signal of light. Okay. So, this light is detected by the detectors that are present, and which then does the base call. Okay. And if you remember, we pass one base at a time. Okay. So, one type of DNTP we give, and we see if there is any signal or not, and this process happens at a very large scale across the picotiter plane. Okay. And that is how we get this high throughput sequencing. Now, one of the first things you probably remember is that we need to amplify the DNA, right. When you prepare the library, we have to do some amplification. In 454, this is the emulsion PCR, which will generate multiple copies of the same DNA molecule on the bead. Okay. And the average signal for all the syntheses that are happening on a single bead, that is detected as pulse of light. So, the assumption is right that all the syntheses in across these molecules in a bead, on a bead, right, they are happening simultaneously, and the signal comes together, right, simultaneously, and that will be detected. We also talked about Illumina sequencing by synthesis method. This method also relies on synthesizing the complementary strand, and here we have reversible termination method. So, there is one by one, right, addition of bases and detection of signal. Again, there is an amplification step required, right. So, what do I mean by amplification is you need to again clonally propagate, right, through bridge amplification, right, and we generate clusters. Again, these are copies of the same molecule. Okay. So, we have seen that there are similarities between these two methods. We have an amplification step for the library before we do the sequencing, and both rely on sequencing by synthesis method. Now, what are the limitations of 454 and Illumina? General limitations. So, one of the things we have seen is that we get short reads. Okay. So, for 454 we get about 500 to 600 base pair reads, whereas for Illumina its maximum is 2 times 250 with the current technology, right. So, this is actually quite short if you think in terms of the genome sizes, right. So, if you are thinking about human genome, this is 3 times 10 to the power 9, right, and compared to that these fragments are very, very small, which means if you want to assemble human genome or such a big genome, you would have to assemble a huge number of reads, right. So, billions of reads will be required before you can do this assembly. Now, there is another problem, is that there are a lot of repetitive regions in our genome and in other genomes as well. Okay. So, if you have very short read, even with paired end sequencing, you will have difficult time actually assembling these repetitive regions correctly. Okay. So, this is one limitation of both these technologies. On top of that, we will have structural variations. So, we will talk about what structural variations are in the next classes. When we talk about variant calling, etc., you will understand that, and especially for resolving the structural variants, these short reads are not really suitable, and we would like to have really long reads, right. So, longer reads will be much better. So, if you compare Sanger sequencing, we have 1 kb, but can we get at least 1 kb, and if we, more than that, right. That is something that would be very helpful for genome assembly. So, as I said, like there is difficulty in assembly of repetitive regions in genomes and identification of structural variants with these very small reads. So, can we get long reads in NGS, right? This is one of the questions that we can ask, and so this what will happen is that if we can get long reads, right, this will actually help in the genome assembly process overall, right. That it will be less challenging because we can then have a lower number of reads to assemble the genome. In addition, we can resolve these repetitive regions quite effectively, and we can also identify the structural variants. Okay. So, today we will talk about one such technology that actually allows us to generate these long reads, okay. And this is called the single molecule real time sequencing or SMRT sequencing in short, and this was developed by Pacific Biosciences, and we get long reads greater than 1 kb, and sometimes we also get reads of 5 or 10 kb length. And the other advantages of this method is that we have signal identification and base call from individual DNA molecules. So, as we go into the principle, you will see, right, instead of these clusters or these the emulsion PCR where you generate a lot of these clones of the of a molecule, right, and then do the signal identification and base calling from the average signal of these clusters. Here we actually identify these bases from a single DNA molecule, right. So, this identification happens from individual DNA molecules. So, which means we have higher resolution, right. And on top of that, we also have real time signal monitoring, right. So, because we are actually following the process of sequencing for individual DNA molecules, we can actually identify when the base addition is happening. If you think about the Roche or Illumina in comparison, right, where we are looking at average signal, we are not following individual base addition events, right, because there will be slight differences in time when this base addition happens in Illumina platform, right, between these different molecules. There will be slight differences, and we have also talked about this phasing problem, right, or we will talk about this phasing problem, pre-phasing etc. later on, and we will see that this will actually lead to degradation of signal later on. Okay. So, when you talk about quality of data, you will see this problem coming up with Illumina data especially. Okay. So, these are two advantages of this method along with the long read that we get. So, the question is now how does this sequencing happen, right? How do we actually get this long reads? So, we will talk about this principle now. So, sequencing is done on something called SMRT cells, okay. Like flow cell, like picotiter plate, here we have these SMRT cells, right, where we can where we do the sequencing. So, it is kind of like an box kind of structure where this sequencing would happen. So, you can look up search and look up for these images, right, and you will find quite a few pictures of these SMRT cells. Okay. So, inside this we have something called zero mode waveguides. Okay. So, one SMRT cell contains millions of these zero mode waveguides, and you can look at this reference to actually see how these zero mode waveguides look like. Okay. So, I will just draw a kind of a schematic of this, right. So, it is kind of like a well, okay, something like this, but the dimensions are so small is that if you shine light from one side, the light cannot pass through the well. Okay. So, what will happen is that only the lower part of these wells are illuminated, okay, and the light will not pass through the well because the dimensions are so small. Okay. And in the SMRT cell, we have millions of such zero mode waveguides. So, you can imagine, right, if you kind of imagine in your mind, right, here you have these and we are looking from top, right. So, and we are looking at all these wells and we have millions of them in the SMRT cell. Okay. So, which means we can do this sequencing for millions of these molecules in parallel inside one SMRT cell. Okay. So, what happens is that sequencing actually happens inside these ZMWs. So, one of the terms I will just introduce is we will see these terms SMRT1M, SMRT8M, you will see this. So, it simply tells us how many ZMWs are present inside the SMRT cell. Okay. So, 1M means 1 million ZMWs per SMRT cell, 8M is 8 million ZMWs per SMRT cell. Okay, so as I shown you, right. So, these sequencing reactions actually happen here inside these wells, okay, and especially, so to be more specific, it actually happens in this region which is illuminated. Okay, so now how does this process occur, right? So, we will go to that in a moment, right. First we talk about library preparation and then we will go into the sequencing part. So, for library preparation we have this genome sequences, right. So, this is the genome, double stranded DNA, right, and the first step is fragmentation. Okay. So, we generate these small fragments from the genome, and this is required because again we cannot sequence the full genome at one go, we need to generate these fragments, but in this case, in case of SMRT sequencing, these fragments could be bigger compared to Illumina or Roche, right, because we can have now reads of 1 kb size or even more, right. So, we can have these bigger fragments generated, but nonetheless we need this fragmentation step. So, once we have this fragmentation, we need to add the adapters, okay. And one of the things you probably will notice later, right, is that this method does not require any amplification, right. So, because we are sequencing single molecules, we do not need to generate clusters like Illumina, right, or the on-beat amplification that we see in 454. Okay. So, the adapters are very special here. So, we have something like hairpin adapters and they kind of look like this, okay. And we can have two different adapters on two sides. Okay, so, it kind of looks like this dumbbell shape, right. So, we have these hairpin adapters. The special part is that you have this double stranded part, right. This is the double stranded part of this adapter and then you have the single stranded part. Okay. So, these are called SMRT bell hairpin adapters, right. So, we kind of get these dumbbell shaped molecules after the adapter addition. So, what actually happens, right, if you kind of get this dumbbell shape, this is the adapter part. Is that in the single stranded part here you can have primer binding sites, right. This is where you can have your primer bind and then sequence the molecules. Okay. So, you can probably now guess is that this is a sequencing by synthesis method. You have the primer, you have polymerase and you have TNTPs which will then synthesize the complementary strand, and by in the synthesis process it will identify the bases. Okay. So, let us now go into the sequencing process itself, right. How does this sequencing actually happening? So, for this synthesis, right, we have DNA polymerase, but we also have special DNTPs. So, in these DNTPs we have fluorophores, right, and each DNTP will have a unique fluorophore, right. So, DATP will have let us say red color and DGTP will have blue, etc., right. So, each type of DNTP will get a unique fluorophore, so which can be then identified by a detector. Okay. So, the fluorescent signal can be detected. Okay. So, these DNTPs their fluorophores are linked to the 5 prime phosphate group of the bases. Okay. And what happens is that during the synthesis process these fluorophores are released, right. The phosphate comes out, the pyrophosphate comes out, right, during the synthesis process, and along with that the fluorophore also comes out. Okay. So, you can now visualize in your mind that this synthesis is happening and the fluorophores are coming one by one, right. So, now let us look at this in much more detail, right. So, as I mentioned, we have the ZMWs, right, and this is where the sequencing is happening, right. The lower part is illuminated and inside this ZMW you have the DNA polymerase sitting at the bottom of the well. Okay. and it then binds to the DNA molecule comes in, right, and of course, you would have to ensure, right, one polymerase gets only one type of DNA molecule, right. This can be again optimized through trial and error. Now, once this DNA molecule comes in here, right, along with the adapters, you can now start the synthesis process. Okay. So, now you remember, right, we have these DNTPs that are labeled, right, some sort of labeling is there, and these are also unique. Right, and F1, F2, F3, F4, these are denoting unique fluorophores, right. These are different fluorophores that can be detected. Now, what happens is that you can imagine you have the synthesis happening. Okay. So, here is your template and here is the primer, right, and we give all these fluorophores together, right, and the polymerase then selects the right one based on the complementary sequence, right. So, let us say you have this addition of A, okay, here, and during this addition of A, the fluorophore will come out, right, it will be released and the detector will detect this fluorescence. Okay. And what will happen next is then, okay, we we then get the next one, right, we get to the next page and maybe next it is G that is attached, right, and this fluorophore will then come out and it will be detected by the optical detector. Okay. So, this is happening one by one, right, you have this addition of base, then you have the release of the fluorophore and detection of signal. Okay. So, this fluorophore gives us the identity of the base. Now, one of the questions you might ask, right, these fluorophores are also present in the well, why do not they give any signal, right. So, these bases are floating around before they are added to the growing strand. Okay. Why do not they give signal? Now, when these bases are free, right, free DNTPs are moving in and out here, right. So, what happens, they are coming in and out, right, randomly very fast. Okay. So, maybe I can draw this separately, right. If you think about this region, we have these DNTPs coming into the illuminated region and going out, right, there this is random motion. So, they are coming in and out very fast. So, the time that they spend in this illuminated region where the fluorescent signal can be detected is very short. Only when they are binding to the growing strand, right, the polymerase holds them for long enough for the detector to detect them. Okay. So, this ensures, right, we are detecting only the bases that are being added to the growing strand. Okay. And this way we can get the signal that is coming out in real time, right. So, you can now see why this is called single molecule sequencing, right, single molecule, because the signal comes from synthesis of the complementary strand of a single DNA molecule, right, that is coming here and binding to the polymerase and where the synthesis is happening. Okay. And in addition, you can see we can detect these signals in real time because the synthesis is happening at the single molecule level and we are observing those individual base addition events. Okay. So, compared to Illumina and 454, this will give us higher resolution, and you will see this kind of real time sequencing will have some other applications. We will discuss about that later on when we actually talk about those applications. Okay. So, I hope this is clear how the sequencing reaction happens. This is sequencing by synthesis. We have the fluorophores being added and fluorescent signal being released, and which is then detected by the optical detector. Okay. So, this detection of base addition happens in real time as I have just mentioned, right. So, if you have the detector, you can probably see, right, you have kind of, if you kind of draw this, right. So, this is time versus signal in the y axis, right. So, detector will then detect, okay, there is this addition of A, again we have different colors, right. So, I am just drawing by one color, right. Maybe then we have addition of T, right. Maybe after certain time we have addition of G and so on, right. So, you are detecting these signals and this data is then also stored by the machine. Okay. So, we can actually retrieve this kind of data and this also can give us some valuable information as we see later on. Okay. Now, when we are detecting this base addition in real time, we can actually generate some of the statistics. Okay. and that we will talk about now. Before we go there, here is a link where you can actually see the animation of the whole process, and you will probably get a really good idea of how this process is happening. So, please look up and see the animation. Okay, so one of the things this method can do is something called strand displacement DNA synthesis. Okay, so if you have used a specific polymerase, you can actually do something like strand displacement DNA synthesis. So what actually is this strand displacement DNA synthesis? Now, as I mentioned, right, so you remember, right, we have this kind of shape, right. This is the adapter here, pin 1 and here. So, I am just now separating them out, right, and you can have this sequencing process going in this direction and going here and also continuing, right. So, you can operate this sequencer in such a way that you get just long reads. Okay. So, you start from here and you end up somewhere here and you get the full sequence of this DNA fragment, right, and this fragment could be quite long, right. You can have 1 kb, 2 kb, etc., and you can get the full sequence. Now, another way you can actually run this sequencer is something called strand displacement DNA synthesis. So, depending on the polymerase, what you can do is that you can simply, so if you consider this as a circular molecule, right, you can simply continue to synthesize these molecule over and over again, right. So, if you just imagine this here, right, and convert this to a circle, right. So, this you can just keep on going synthesizing the same molecule over and over again. Okay. Why would you do that? So, we will talk about that in a moment, right, why we want to do this, right, why do you want to sequence the same molecule multiple times. Okay. So, but there is this capability where you can actually do this strand displacement DNA synthesis, so that you go over the same sequence multiple times. Okay, so, there are two parameters. Since you are observing the signals in real time, there are two parameters that you can measure, and again these will actually have some implication as we will see later on. So, one is called interpulse duration. Okay. So, this is the signal that we are detecting, if you can imagine, right. Again we have time and we have signal, and let us say we are detecting these different signals, right. So, you can imagine this is what is happening in real time, and let us say this is A, maybe this is T, this is also T, this is also G, right. So, maybe this is what is happening. So, one of the parameters that you can measure is the time between two pulses or two signals. Okay. And you might ask, right, what is the importance of these? Okay. As you will see, we can use this signal for specific applications. This specific time difference actually will tell us something about the DNA. Okay. So, we will talk about that later, but this is something that is quite important. We can measure this interpulse duration because we are observing this sequencing process in real time. Okay. There is another parameter that we can also measure is something called pulse width. Okay. So, this is again if we draw the time versus signal, so this is what is pulse width. So, what you will observe, right, if you kind of look at the real data, you will observe these pulses of different widths. Okay. And again the question is why is this important, right. So, I will tell you that this kind of signals can be then combined with interpulse duration, and it can help us understand the modifications on DNA better. Okay. So, we will talk about this in much more detail later on, but just to be aware that these parameters can be measured, right, and can be stored in the data, which will be very useful for different types of analysis. So, there are two parameters we have talked about, one is interpulse duration and other is pulse width. Okay. So, I hope this is clear, right, what interpulse duration and pulse width is. Okay. Now, what are the advantages of SMRT sequencing? So, the first advantage is the long reads that you get, okay, and this is something that kind of comes out favorably for this method compared to Illumina or 454, right. So, you get really long reads, and that kind of will be helpful for genome assembly process. and we get average read length of 2-3 kb, and some reads may be quite big, right, 10-20 kb, but at least we get more than 1 kb, right. So, which is more than double the double of the existing sequencing technologies. We have single molecule resolutions, right. So, we have seen we are getting this data from single DNA molecule. We are synthesizing the complementary strand of a single DNA molecule, and this process is happening across multiple wells, right, multiple ZMWs, right. We have discussed that, and millions of these ZMWs, right, we are getting parallel signal, which will give us sequence for millions of molecules, but at single molecule resolution. So, it is incredibly powerful. And we also have observation of events in real time, and we have seen that we can measure these two parameters, interpulse duration and pulse width, which will have some applications that we will talk about later. What about the drawbacks? So, as we have seen, we require enzymes and modified nucleotides. So, it means the running cost would be a bit high, right, but probably very similar to Illumina. Again, we have in Illumina we have modified nucleotides and polymerase. The major drawback is that it has higher error rate, which is about 14%. So, this again varies, this is something that has been measured, but it is significantly higher than Illumina. Okay. And why is that the case? So, what we think is because of this polymerase being exposed to light, it kind of affects the proofreading activity of the polymerase. So, to avoid to circumvent this issue of higher error rate, there is a method that has been developed called the circular consensus sequence, or CCS. Okay. So, what actually happens is that we get something called the strand displacement synthesis. So, we do this strand displacement synthesis and we get something called hi-fi reads. Okay. So, what is hi-fi? So, this is high fidelity reads. Okay. So, as I have just mentioned, we do strand displacement. So, what is happening is we are sequencing the same molecule over and over again, right. So, we are getting just data from this, right. We are getting such like for the same region we are getting this multiple reads mapping to the same region, and these will reduce the error rate, right. So, you can imagine if you are sequencing the same region multiple times, and in one call we have an error here, in another call we see a mismatch here, right, compared to the reference sequence. So, we can say, right, okay, these two are probably sequencing errors, because if it is a true mutation, it should be appearing in all the reads that we get, right. So, we are kind of sequencing the same region over and over again, and we are calling the consensus, right. So, all the errors can be then taken care of by this process. Okay. So, again here is a link where you can probably see how this method works, and you will get a better idea of this through the animation. And the advantage is that hi-fi reads are now long and as well as they are accurate. Okay. So, this is something that is kind of a really game changing, because what you have seen so far, right, the short reads sequencing methods, they are very accurate, right. So, that is good, but they have some drawbacks. Okay. And to counter that, this long read sequencing was developed, but it had higher error rate. Okay. And to circumvent that, if you can then make sequence long reads, right, and also get really accurate data, that is the ideal situation, right. So, this is something that is very helpful and useful for all sorts of purposes. Okay. So, just to give you some idea, right. So, how many reads you get, number of hi-fi reads that you can get for this SMRT cell. So, this is from Pacific Biosciences specifically. So, you have SQL2E, SQL2 systems, these are the name of the sequencers, and the SMRT cell that you can use, and SQL2E and SQL2, these are the recent the modern ones, and you can get up to 4 million reads from them. So, here are the references that you have used for this class. And to summarize, we have introduced single molecule real time sequencing, which is based on detection of fluorescence signal from the synthesis of DNA that is happening inside ZMWs, right. So, these are incredibly small wells where the polymerase is immobilized and the synthesis process happens. So, this is something that is actually also in contrast with the 454 and Illumina, right. So, you have seen in both those methods the DNA is immobilized on picotitor plate or on the flow cell, and the polymerase comes and it does the synthesis. Here in SMRT, it is actually the opposite, where the polymerase is immobilized at the bottom of the wells, and the DNA molecule comes and the sequencing reaction happens. And what we have seen, this allows us to observe sequencing in real time and at the level of single molecules, which actually have some real benefit for different applications, and we will talk about that later. And from these real time observations, we can derive certain parameters, which we also talked about, and we can utilize those parameters for some applications. And what we have introduced also is that the original long read sequencing actually has very high error rate, whereas the circular consensus reads, the CCS reads, they actually deliver long read data, but also with high accuracy, and these are called hi-fi reads or high fidelity reads, and these will have very important applications in genome analysis. Thank you.